Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Jordan Stillman, Digital Products and Marketing Coordinator for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. Please enter your questions at any time. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Dr. Anna Bris from Thermo Fisher Scientific.
Thank you for this introduction. So today I will talk to you about the antibody purification solution from Thermo Fisher. As you may know, Thermo Fisher offers premium solutions for the whole process, BioProcess, from the DSP and the USP through the analytics. In purification, we offer two lines of resins. They are called porous chromatography resins and Capture Select Affinity. The Capture Select resins are the largest portfolio of affinity resin, which are dedicated for the purification of antibody formats, but also biosimilars and viral vectors. The porous resins offers also ion exchange and HIC resins on top of that. We have a state-of-the-art manufacturing facilities with large-scale manufacturing commercial lot size up to 250 liters for porous and up to 15,000 liters for the Capture Select lines. Besides the standard portfolio, we also offer a program for the development of custom resins if you have specific targets which are not being addressed on the market right now. As you may know, there is a growing diversity of antibody molecules format in development on the market right now. As of now, there is around 2,500 molecules in the pipeline, which is 12% rise over last year, and that brings us to 31% of market share right now. For the pandemic response with the COVID, we also saw a tremendous increase on the project on antibody molecule. There are a lot that have been approved already and quite a lot that are in phase 3 to phase 3 trial, as well as preclinical stages. That is, of course, very important, and the thing which is different than the years before is that it is not only addressing standard antibody format, but also novel and different format. And these different formats are the ones that are listed here, so it can be bispecific, Fc fusion protein, or also Fc fragments. As you know, the affinity chromatography has always been a very big advantage for the antibody purification. Indeed, with an affinity chromatography, instead of several ion exchange steps, for example, you can reduce tremendously the number of purification steps, and by this way, you can increase your product yield while decreasing your process development time. And this leads to a reduced time to market and a better cost of goods. You may know this graph on the left, which shows that, for example, if I take the light blue line, you have 85% of yield for every step in your process. And even if it's good, if you have several steps at this specific yield, in the end, after 4 steps, you would have only 50% of your molecule left. So the best is, of course, to reduce the number of steps, and the affinity chromatography is the right candidate for that. Historically speaking, the Protein A has been a very nice solution for this particular affinity purification, and it's an established purification platform now for all standard human IgG-based therapeutics. It binds very typically between the CH2 and CH3 domain of the antibody, and it has proven to be very efficient. So nowadays, there is not only standard antibodies on the market, but also novel formats, which are not obviously completely being purified the right way with Protein A. These new formats are, beside the typical IgG, bispecific antibodies, for example, or Fc fusion proteins, or also fragment Fab. And the thing with this novel format is that sometimes they just don't present the typical Protein A binding site, which is CH2, CH3, or it's an altered binding site. Some of them, like bispecific antibodies, can be also very pH sensitive, so they could aggregate a certain pH or lower pHs, or some other modalities are known to produce higher level of aggregates and formate dimers. That is also a fact that sometimes you have the high and low molecular weight species that are not addressed or removed through this single step. And not the least, the last but not the least, you have also light chains, sometimes free and light chain dimers that can just be in a large amount in your sample. So for these, we have developed by Thermo Fisher a portfolio of 4 different resins that can address these different novel antibody formats. These are these 4 resins that are listed here. We have, for example, the Capture Select CH1 XL, and as the name indicates, it binds the CH1 domain here on the green. We also have a Kappa XP resin, which will bind any Kappa domain of your antibody, as well as a very new resin, which is a red on the bottom of the slide, which is Lambda XP, which will bind lambda domain of the immunoglobulin. And we have for the yellow part here, which is showing the CH3 domain of the antibody, a resin called XP that binds specifically to the CH3 domain. All these resins present the advantage to be very specific and not binding the typical CH2, CH3 interface of the antibody, and also to offer the possibility to elute with mild pHs, not that acidic as a protein A. Just some words about the Capture Select technology. So we use for that targets that we immunize llama with, and they produce what we call this camelid IG. These camelid IG are very smaller and very more stable than the typical antibody, and they can then support more aggressive condition and more different condition too. This is produced through yeast. That means that you get a product which is completely animal origin free. And because it's very convenient and standing different pHs and harsh conditions, you can use that in commercial purification processes. And it is already in several clinical trials and phase 3 processes around the world. I will go a bit more to details for each of the 4 resins I just mentioned before. So let's start with the CH1 XL. As the name indicates, it binds a constant heavy domain CH1 of all human IgG subclasses. There is also no co-purification of free light chains, which is a big advantage. The binding capacity is around 19-20 milligram per milliliter for a polyclonal Fab, and you can use a mild pH to elute, so around 4 to 5, not obviously 2.5 or 3. It's also available on magnetic agarose beads for automated workflows if needed. The specificity is very high. So if you look on this SDS page on the bottom left, you would see that load was very, very turbid and very charged in contaminants. And the elution, the last row, the last well, you show very high purity. It's like 95% purity. And this is a feed from HEC 293 cells. And we get the same thing from different feed from CHO, for example. When I said that it would not bind free light chain, this is to be shown on the right graph where a lab chip analysis is showing on A, the load before the purification. So we have LC, LC2, which are free light chains and light chain dimers. And on B, that's the eluted sample, which is purified, and there is an absence of these free light chains. Let's go to the Kappa XP. Resin, which is binding the Kappa subtype of all immunoglobulins containing a Kappa light chain. The capacity is quite high. It ranges from 20 to 45 grams per liter, depending on the format. And the elution can be made at mild pH 2 up to pH 6. So it's quite interesting when you have molecules that are sensitive to lower and acidic pHs and tend to aggregate. On the below graph on the left side, you can see that at different resonance time, but a typical resonance time of 2 minutes, the capacity as 10% breakthrough is around 40 grams per liter. At 5 minutes, 4 minutes, we would be a bit 44, 45 grams per liter. It is true that the resonance time has an effect, but you can have a quite high capacity. And the purity is also very good. And if you see on the right side, on the rituximab, the purity here shows on the well 3 that one-step purification gives you only intact IgG and light chain deletion fraction. So it's a quite straightforward resin to purify your kappa light chain containing antibody. Let's go to FCXP. FCXP is a preferred resin for all antibody which have an altered or absent CH2, CH3 domain, so that cannot bind protein A. FCXP binds exclusively on the CH3 domain, on the apex of the CH3 domain for any kind of human IgG. The capacity is very high. It is usually above 40 grams per liter, and you can also elute at mild pHs 4 to 4.5. As you see on the residence time panel below, there is a 44 milligram per milliliter capacity at 4 minutes residence time. You can go higher if you increase the residence time, of course. That is also for the rituximab gels on the right. You can see the purity of it. And the thing that this one is very much liked and used actually already in commercial or phase 3 and clinical phases processes on very large columns that are above 200 liter because of this advantages that you can bind even if the protein A binding domain is not available. It also allows you to have a very high purity in one step, so you can reduce the step of your processes. And the last one, which we will be launching mid of August, and it's called Lambda XP, is binding as the kappa XP, the lambda subtype and light chain for all immunoglobulins here. The capacity is also quite high, 35 grams per liter, and you can elute at pH 3.5 up to 4. Data are being generated right now, but I can show you the binding capacity at 4 minutes residence time below on the left, and you can see that it's around 40 grams per liter. The elution is very efficient, and you can see on the right side, after the flow through, a quite narrow peak for the elution. It's here bispecific antibodies at pH 3.6, and the load concentration was 32 milligrams per milliliter. The peak is very narrow and very high, and the elution is just very efficient. Beside these bulk resin that we give, of course, we have also pre-packed solutions for you, and that's our portfolio of Evolve D columns. Which key features are listed there. They're, of course, prepared and packed in a classified ISO 7 environment, meaning that they are ready to use, plug and play, to be used in cGMP processes. And they are, of course, suitable for direct connection to any chromatography system you might have inside. They also have a non-metallic flow path that eliminates the corrosion risk if you are using high salt concentration in your process. They are given with all the regulatory support documents which are available on the hardware and the packed resin. We propose 2 bed heights, as you can see on the right side, 10 and 20 centimeters, as well as different column diameters, 7, 10, and 20, which offers you 6 different sizes ranging from 385 milliliter to almost 7 liter. So it's a nice way to screen resin and to start to work without having to care about the packing. Beside all this capture resin that I just described, we have also another resin which is used to polish, and this is especially to remove HCP. As you may know, there is a lot of HCP coming from CHO-produced mAb, for example, and bispecific mAb, and the list is here to be seen with catepsin, clusterin, G-protein carpal receptors, and etc. The clusterin is usually presenting quite amount in your sample, and we have this resin which we call Capture Select Clusterin Clear, and this has been reported to remove in one shot in flow-through application up to 95% of the clusterin, but not only, also 65% of the other top HCP that are usually being seen in this kind of processes. We have also an interest on other kind of HCP removal resin, and we are developing right now 2 others, which are the lipoprotein lipase and phospholipase. That's my last point on the bottom right of the slide. If I would summarize now, the advantages of these resins to address the purification challenges of novel and different antibody formats, I would say that they provide, through their unique specificity, through their production technology, a high purity and a high yield in a single capture step. They also, because of that, help to reduce the number of process steps. And we have examples of people that could remove 2 or 3 steps just by using one of these resin. They are, of course, completely upscalable. So you can do that in GMP, big, large production columns, above 100 liter column volume. And we have some pollution solution, like I show with the clustering. If you are going to test or use that, be aware that we offer complete technical support behind and services to help you take the best out of the resin and develop the best protocol with that, so that you would really take the full advantages of this technology. And with that, I would like to thank you for your attention. I'm happy to answer your questions. Thank you.
Thanks, Anna. So we do have some questions coming in. The first question I see here is, can you explain about the CGMP compliance?
Oh, yeah, sure. That's a legitimate question. What we mean by CGMP compliance is actually these resin are completely validated to be used in GMP environment. So there is a validated production processes, and there is no validation objection on any kind of steps during the way. We have also a complete regulatory support package that is available for you if needed. All GMP requirements are being respected, and when they are launched as a GMP resin, they can be used for commercial processes.
Fantastic. And what are the scaling up capabilities and market adoption so far?
Yeah, that's also a question that comes on and on. It's very widely adopted right now because they are GMP and because they are quite unique. This technology is the only one on the market so far. These capture select resins, not only for antibodies but also for biosimilars or other kind of proteins, are being used in clinical and commercial processes already on manufacturing. We have example like with FCXL by large customers using that in a large scale, like 200 liter column for biospecific, and it helps them, for example, to remove one process step in the whole process and to gain place in the facility. They could not afford to have more place. So this is something which is usually playing a big role, and owing to that, we are in quite a lot of commercial processes right now. They are scalable. They are not difficult to pack, and they can be used in very large columns.
Thank you. And here's another question. How do you ensure security of supply for the affinity resins?
That's a good question. We have scale capabilities available for ligand manufacturing with free non-occupied sites for the manufacturing. So we have, for example, a manufacturing process partially redundant in other sites. So we have a lot of capability here. So we are not limited right now, and we have some buffers, so to say, to produce even more than what we sell now. So there is so far no risk to have a short supply coming in or an issue of supplies coming in.
Thank you. And what are the cleaning recommendations for the resins?
That's quite classical. They are quite not sensitive to acid, so we can use, it's depending on the resin, but most of the time we can use different type of acids. We can use urea, we can use guanidine in different concentrations. They can be quite clean harshly. So that is not an issue there. And we can also use NaOH in specific concentration. Most of the time it's around 25 millimolar NaOH. And depending on the resin, we can use, for example, up to 6 molar urea or 2 molar guanidine. So there is a complete set of buffer that you can use. And when the question comes and you have to develop a cleaning protocol, I can help, or my colleagues can help from the technical support to find the best condition for you. Most of the time, 1, 2, sometimes 3 steps, but 2 steps are efficient and enough for you to get a quite clean resin to reuse.
Perfect. Thank you. What are the main advantages against protein A resins?
Yeah, that's a good question. I would say that it's a unique specificity offered here. So it's very dedicated to a specific target. That's a good example is when you have a custom target. You have your target, we just immunize the llama, and we get antibodies from the llama which are only directed to this target. So it allows actually to have an early removal of unspecific species very early on. That's why you get, as you could see during the call with the STS page, you could get a very high purity in only one step. So what stays there are very similar or aggregates or dimers from your molecule, but the purity is very high, and that whatever the kind of target you have, that is quite unique. A big advantage too over protein A is the mild pH for elution. This is a big advantage to avoid aggregation or formation of dimers. The fact that you can go up to 5 or 6 sometimes to elute with the pH allows you to have less worries on the stability of the molecule. And also, it's a bit different than not only protein A, but also protein L. So you have, for example, the CH1XL is a kind of good alternative to protein L if for whatever reason it doesn't work for you. Another big advantage is also that it comes from camelid, and this VHH molecule from camelid proves to have no immunogenicity when you put it in a human body later. So it's actually most of the time safer as protein A. It's less dangerous, so to say. I would say that's the main advantage. So the purity in one shot, the high unique specificity, the mild pH allowing more stability, and this lack of immunogenicity.
All right, and I think we have time for just one more question. What tools are available to support the resin screening?
Okay, yeah, sure. You have, as I say, we have that in bulk, so you can pack yourself. You have also this pre-packed column that I presented, the Evolved, but they're quite big. So what we have as another offering, if you want to do a quick screening and compare with other kind of column sizes, we have pre-packed the small pre-packed column and also rubber columns if you want to use with a robot like Tecan. We also have, for example, CH1XL, which is available on magnetic beads, which is interesting in some cases. And beside that, we also have, because you would have to use that, kits, ELISA kits that would allow you to determine the leakage of the resin. We also have some ligands which are available not as ligand onto a classical resin, but biotinylated ligand. If you want to check that with a streptavidin assay. So there is this kind of solution that you could use if you need to screen very fast. The robocolumns are very appreciated usually. And anyway, you could also use bulk in 96 well plates.
All right. Well, thank you so much for being with us today, Anna.
Thank you. And any questions that have not been answered can be reached. We will reach out to you anyway after the call.
Fantastic. And thanks to our audience for joining us as well. The recorded version of this webcast will be available for on-demand viewing on our website. And as a registered attendee, you will receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.